We've actually discussed this already some because we have the aminotransferases or the transamination reactions, and that utilized PLP. PLP also did racemizations, and what was the third class of types of reactions that pyridoxal phosphate did? Decarboxylation, which those of you who are currently in neurochemistry, we've actually discussed some of this already, and so don't ruin the surprise for some of the others. I'm sure we'll keep them on their toes. All right, another type of reaction that occurs frequently in biosynthesis reactions are one-carbon transfers. So we're talking about methylation in particular. Okay, and so the two cofactors are tetrahydrofolate, which is one of the reasons why you need your folic acid, especially if you're a female. And then S-adenosyl methionine, which what's that one more commonly called? Does anyone know? What do they call it? SAM, S-A-M, S-adenosyl methionine. And so yeah, it's the same cofactor. And then we can also have amino transfers from glutamine. Okay. So glutamine can become glutamate and can give off that nitrogen to whatever needs the nitrogen at that point. Okay? And there are many reactions that use glutamine as a major source. Okay? So not only for other amino acids, but also for nucleotides in particular, since those are the nitrogenous biomolecules. So we're going to actually look a little bit in greater detail about the way that glutamine is used. Because what was the functional group in the end of the glutamine? What's that functional group? It's an amide, or an amide, right? And so it becomes glutamate, but what's the ramification of it losing an NH3 group? What would be bad? Like, what's so bad about NH3? It's toxic. Remember, it messes up with the osmosis in the brain, causes glutamine deprivation, swelling, death. And so you don't want that nitrogen to get loose. And so there is a way around that enzymatically. It never leaves, even though the nitrogen is removed, it never leaves the enzyme. It goes through a special little channel. Okay? So these are called glutamine amido transferases. And it transfers the amino group, so that's where you have to be kind of careful. Because remember, glutamate is the one that's used in all of the amino transferases. Glutamine is for the amido transferases. There are two domains. One is going to bind the glutamine, and the other one's going to bind whatever's going to get the nitrogen. And there's a tunnel, for lack of a better word, that connects the two, and that's what the ammonia goes through. It never leaves that channel. Okay, so you don't have to worry about ammonia escaping and messing up the pH of your body or causing some type of hydration within your brain. And it's a conserved cysteine that's a nucleophile. Why? What is it on the cysteine that can be a nucleophile? The sulfur, the thiol. So this is actually really quite simple, and it relates to what we've talked about in organic chemistry 2 with amide reactions, right? Because you make tetrahedral intermediate, you collapse, that kind of thing. Just what we see before. Okay, so the most confusing thing about this figure from your book is the substrate here. See, it says ROX. That's because it can be from an alcohol, or it can be from like a ketone or an aldehyde. Okay, and so, but if you're going to be an alcohol, you have to activate it somehow first, because hydroxide by itself isn't a good leaving group. Whereas if it's phosphorylated or something like that, or adenylated, then it's much better. Okay. All right. And so it binds the glutamine. Let me just do it this way. It's easier for me to write on it. It binds the glutamine within the glutamine binding site. And then we have that cysteine that acts as a nucleophile. Okay? And just like what we've seen in the past, in organic chemistry, what's it going to smack into? The carbon of the carbonyl. You form, it's not showing it here, but you form that tetrahedral intermediate. And then every time we form the tetrahedral intermediate, what's the second step of the reaction mechanism? Collapse. Something has to break or leave. And one is it could go backwards. That's why it's reversible. But usually what we're talking about is it has a general acid nearby, and so it's the ammonia that leaves. But once again, it just goes through this channel to the second binding site, where there's already either an activated alcohol group waiting or a ketone aldehyde. Okay. And as you know from organic chemistry too, if you start off with a carbonyl group and nitrogen does the attacking, you end up with an imine. If you started off with the alcohol, you're going to end up with an amine. Okay. But yeah, it's completely analogous to what we've seen in organic chemistry too, which is nice to see why organic chemistry is relevant, biomedically at least. Are there any questions about this? There's two binding sites. There's the ammonia channel in between, so that way we don't have to worry about the deleterious effects of ammonia.
Did you say imine and
Imine
amine?
and the amine, yeah. So if it starts off here of the carbonyl, since that nitrogen is going to have its own pair of electrons and hydrogens that can pop off, it can attack it twice, and so it forms an imine, which is a carbon double bonded to a nitrogen. If it starts off with the alcohol, then that phosphate is a great leaving group, and so you just end up, it can only attack one time, so it's an amine, an NH2 group or an H3 group, depending upon the pH. Okay, and so that was just the end from what I was hoping to get to on Wednesday. And then we're going to just pick up right in the second section of this chapter. Okay. So now we're going to start talking about the biosynthesis of specific amino acids. Because do we make all the amino acids in our body? No. So we only make what are called the non-essential amino acids, but there are several essential amino acids that we have to get from our diet. Whereas plants and bacteria often are able to make all 20 naturally occurring amino acids for protein synthesis. Okay? So mammals can only synthesize 11. So there are nine essential and 11 non-essential amino acids. All the others must come from your diet. And unless otherwise noted, all the mechanisms and stuff that we're talking about is a bacterial biosynthesis. There are some times, it's really important, like when we talk about methionine and cysteine and things like that, where there's a difference between the two, between the way that bacteria do it and the way that mammals do it. Why would that be important? Why is it important to know the difference between, say, mammals and bacteria? It's antibiotics. A lot of these then are, like what I did for my research, where if you can find the differences in the mechanism, then you can have a good antibiotic. Hopefully a good antibiotic. Because then you can knock out the E. coli or the Pseudomonas and, and not kill off the human.
That's always good.
That's always a good one. Okay, all the amino acids are made from derivatives from either glycolysis, citric acid cycle, or the pentose phosphate pathway. Okay. The one pseudo exception is arginine in bacteria. Well, technically, we can also do this arginine because we actually already know how to make arginine from something. Where have we seen arginine that was not used in a protein? It's in one of the other metabolic pathways. Well, no, I mean, what pathway is it? Urea. Like arginine, yeah, it's right there and with arginase, and so you can get it from the urea cycle. But that's actually not usually the way that, when we have low arginine, that mammals do it, but that is the way that bacteria can do it. Okay, some of these are within just one or a few steps of what's called the parent amino acid. So for example, I would already expect you to know how to make aspartate from oxaloacetate. Right? Because what is the one enzyme that is used to go from OAA to aspartate? Well, that's the substrate, but it's aminotransferase is the enzyme. Mm-hmm. Okay. So many times they organize these according to the metabolic precursor that it comes from, and I'll talk a little bit more about PRPP in just a moment. So there are those that come directly from alpha-ketoglutarate, which we've actually already talked about glutamate and glutamine. You know, glutamate is just the alpha amino acid form of the alpha-keto acid, alpha-ketoglutarate. We know that glutamine can be, because you can aminate glutamate. I think that's kind of like hip-hoppy. And then we're going to actually talk about proline and arginine. But then there are those that come from the, that are like glycolytic intermediates, so like 3-phosphoglycerate makes serine, glycine, and cysteine, so on and so forth. But this PRPP, which is 5-phosphoribosyl 1-pyrophosphate, it's common in many amino acids and nucleotide biosynthesis, and it's derived from the pentose phosphate pathway. But like I said, we're going to go into greater detail with it. I'll blow it up so that way you can see what's going on. Okay. So it's derived from the pentose phosphate pathway, which we saw back whenever we were talking about glycolysis and gluconeogenesis. Remember, we have the glucose 6-phosphate that can go off one direction, well, go off in all three of those directions, depending upon what your body needs. But whenever your body needs to make new nucleotides or certain amino acids, then you're going to undergo pentose phosphate pathway. Okay. And so this is the enzyme's reaction down here. The ribose 5-phosphate requires ATP, and you make the 5-PRPP, because I'm going to get tired of calling it everything, and AMP. So what class or classes of enzymes can do this? Possibly a synthetase. What else could it be? A kinase. And this one's a kinase. It's ribose phosphate pyrophosphate kinase, which is a mouthful. And it's, you know, the first step in making a lot of these amino acids. Now, on your own, you'll want to review how nitrogen enters in pathways, and once again, it comes from glutamate and glutamine. It's from earlier in this chapter, and also from whenever we talked about how to get rid of nitrogen as well. So I'll let you look at those on your own. I do expect you to know how alpha-ketoglutarate forms glutamate and glutamine. Okay, you already talked about the enzymes involved, so like the aminotransferases. We've talked about the synthetases before, so we don't have to worry about those. That way we can focus on proline and arginine, because those are new. This is the big picture. Don't worry, we're gonna blow it up into individual. But proline and arginine, most people don't realize that they are related. You know, they look nothing alike, but they come from the same ancestral beginnings, I guess, of the biochemical pathway, metabolic pathway. So it starts with glutamate. So if we just look at this first step here, it utilizes an ATP to make what's called gamma, which just means it's hanging off the end of carbon, gamma glutamyl phosphate. So what class or classes of enzymes could this be? It's a kinase. If you're going to name this one, what would you specifically name it? What are you kinasing? Glutamate. So it would be glutamate kinase. See? Oh, yeah. So you guys have already learned a lot. And so one of the reasons why we do this is just because you are activating that. Phosphates are great leaving groups, and so you get to activate that end in order to turn it into an aldehyde. Because it's so hard. If you look at it before, under physiological pH, this is a carboxylate. And so for that oxygen to leave, it would have to leave as an O2 minus, which would never, ever happen. And so by putting it in as a phosphate easily leaves. It's a great leaving group. So this utilizes an NADH or an NADPH, kicks off the inorganic phosphate to make an aldehyde. So what class or classes of enzymes utilize NADH or NADPH? Reductases. What else are they sometimes called? Dehydrogenases. And so if you were going to name this one, what would you name it? This one's glutamate, but if you say glutamyl phosphate, I would definitely give you the credits for it because people will know specifically exactly what you're talking about. The next step, if you remember from organic chemistry 2, is cyclic reactions are usually non-enzymatic. They happen on their own because we have both a great nucleophile and a great, so we got a great nucleophile and a great electrophile, so the nitrogen can come in and smack the carbon, and you form the cyclic imine, which is called a Proline. So that one doesn't even require, it just naturally happens. Just like your sugars can be linear and cyclical. Now the very last step requires NADH or NADPH. So what class or classes of enzymes could this be? Dehydrogenase? Okay. So what would you call this one? Right. This one actually is a reductase, but if you called it the dehydrogenase, I'd give it to you because, once again, people know exactly what you were talking about. It's just a historical name for the reductase.
So if you were to say like the P5C.
Yes. Mm-hmm. Absolutely. So that's how we make proline. Arginine takes it's a little bit more tricky. This is the arginine being made in bacteria. And technically we can make it this way, but it just wouldn't be, usually under the conditions where you need arginine, it wouldn't be, energetically feasible. So this first step, which it's frozen now. Okay, we have go from glutamate to N-acetyl glutamate. So we've made an amide formation by using coenzyme A, our good friend coenzyme A. So what class or classes of enzymes can do this? Maybe an acetyltransferase or a synthase. And so if you were going to name this one, what would you name it? This was the acetylglutamate synthase. Then if you notice, the next two steps are very similar to what we've seen with proline. So they're side by side. So we take this N-acetylglutamate. We're going to add a phosphate at the expense of ATP in order to activate that terminal carboxylate. So what class or classes of enzymes could this one be? Kinase. And if you were going to name it, you would call it? Yep. Yeah, N is just a locant for those who, it's been a while since Organic Chem 2. All right. Then we have NADPH or NADH being used in order to kick off the inorganic phosphate. So what class or classes of enzymes could this one be? The dehydrogenase. And if you want to name it, what would you name it? It may be a mouthful.
N-acetylglutamate, gamma.
Well, N-acetylglutamate dehydrogenase. But if you wrote out N-acetyl gamma glutamyl phosphate dehydrogenase, yeah, I would give you the points for that. Yeah, well, everyone in the field would know exactly what you're talking about because you say what the substrate was, the enzyme's family name, and everything. Now we're going to make acetyl ornithine. Well, we're not. The bacterium will be making acetyl ornithine.
Thanks for the clarification.
Thanks. Most of us aren't too buggy. Be here all week. No. And so what class or classes of enzymes could this be? Pardon? This is an aminotransferase. If you want to be very specific, it would be the N-acetylglutamate, semialdehyde aminotransferase. But yeah, it's a mouthful. But yeah, it's just an aminotransferase. So we've seen that one over and over and over again. We're not quite. However, you should start to see how the arginine is forming along and how it's going to play a role with the urea cycle. Because now we take N-acetyl ornithine and we remove the acetyl group. So we deacetylate it. And this one's got a special name. So you know those that have the special names where we've hydrolyzed or we've broken them apart, like argininosuccinase, so on and so forth. This is the N-acetyl ornithinase. Yeah, that one. And then from here on out, it's just the urea cycle. Okay, so it's the way for the bacterium in order to utilize those enzymes of the urea cycle. So the first step was what we had called like step 0 or step -1. No, I guess that one we were calling step 0 of the urea cycle, where carbamoyl phosphate carbamoylates it. And so does anyone remember what that one was called? You're on the right track. Ornithine carbamoyl transferase. Yes. But at least it tells you, like, if I gave you that enzyme name, you should be able to specifically say, hey, it's transferring a carbamoyl group to ornithine. Then we have, in the urea chapter, they divided this next step up into part A and part B, but this time they're just showing it as one complete step, where we had to utilize aspartate and ATP in order to make argininosuccinate. So what class or classes of enzymes would this be? The synthetase, since it required the ATP, it required that external energy. So what was the name for this one? That's right, argininosuccinate synthetase. And then finally, you kick off fumarate, which where does the fumarate go? This is a good review for the final. Citric acid cycle. And we now have our arginine. And so what was this last enzyme called? This was argininosuccinase. Argininosuccinase, because the arginase would be, if you're in the urea cycle, would be going on to kick off the urea, and that's called arginase. Because a lot of times these that have those special names are whenever we're hydrolyzing it, like either hydrolyzing or forming like enolase and aldolase and argininosuccinase and ornithinase. However, when we are really needing to make arginine, so we mammals, we actually scavenge it from proline since they were very similar. And so this goes back to that P5C, that next to last step from proline. Okay? Because chances are, if you're low in arginine, you're not going to be able to, it doesn't make sense to try to make it from the urea cycle. Okay? Because if you're under urea cycle conditions, chances are you don't need arginine. So, this one utilizes an enzyme that goes frontwards and backwards, okay, at the expense of an alpha-ketoglutamate. And what it does is it takes the P5C from the proline biosynthetic pathway and transforms it into ornithine. Okay, so what class or classes of enzymes have we seen before that does this kind of thing? It utilizes an alpha-ketoglutarate and a glutamate as the other ping to the pong or pong to the ping. It's an aminotransferase. This is ornithine aminotransferase. Except, why do you suppose it says delta? What does the delta mean? It's in the 4th position because all the other aminotransferases that we were talking about before were alpha aminotransferases. This one does it. It recognizes the rear end, so to speak, of ornithine, the alpha, beta, gamma, delta position. Okay, and so that's why, but it is ornithine aminotransferase. It's just not doing it at the alpha position, since ornithine is already an alpha amino acid. Okay, and then from here, from ornithine, now you can go on and to make your arginine. Okay. All right. So the next group of amino acids are going to come from 3-phosphoglycerate, and that's serine, glycine, and cysteine. Okay. And serine, glycine, and cysteine. So, and we're going to see how some of these are utilized later on to do like miraculous things. Like I always marvel at what glycine does, because it's so little and tiny and seems so insignificant, yet it makes a huge molecule in your body. It's the building block. Okay, but where does the 3-phosphoglycerate come from? We had to scavenge it from somewhere to make the serine. It's glycolysis. This is where we really need to pay attention to the cofactors, because we're going to see some new ones here, like tetrahydrofolates coming up. Okay, so in this first step we have 3-phosphoglycerate. We take an NAD and we make it into an NADH. Where's one of the possible places for the NADH to go? It can go to the electron transport chain, so you can actually utilize this to make some energy, at least at this step. Okay, and that's going to make 3-phosphohydroxypyruvate. So what class or classes of enzymes could this be? This is dehydrogenase, and so if you were going to name it, you would name it
3-phosphoglycerate dehydrogenase.
Right. Now we take the 3-phosphohydroxypyruvate, and we actually are going to transform it from a keto acid, which is what it currently is, into an amino acid. It's just not the amino acid that is used to build proteins. It's one that's used usually for regulation. And so it takes a glutamate and transforms the glutamate to an alpha-ketoglutarate. So what would this be? This is the aminotransferase. What specific aminotransferase would it be? Well, it's even more specific.
Glutamine.
It's the phosphoserine aminotransferase. Right. Because there's another serine aminotransferase, but it doesn't require it to be phosphorylated. Okay. So this one's very specific. And so now we have the phosphoserine, and we're going to use a water molecule, so the oxygen then can just act as a nucleophile, and it kicks off that great leaving group. The phosphate is a great leaving group. So what class or classes of enzymes could this be? Phosphatase. Oh, yes. Remember, what was the difference with the phosphatase versus a kinase? It doesn't require ATP, and you get an inorganic phosphate. Just like a pyrophosphate. It's been a while since we've seen phosphatase. Just like the pyrophosphatase, you get an inorganic pyrophosphate without using ATP, or any of the NTPs, I should say. So that's how we get serine. It's just one additional step from serine to glycine. This one is interesting. This is tetrahydrofolate, which comes from folic acid, and it gets transformed into a methylene tetrahydrofolate. So the only difference, let's see, how can I explain this? The only difference between a serine and a glycine is what? CH2OH. What can you call a CH2OH? Well, it's close to methanol, right? Methanol would be CH3OH. Okay, and the reason why is because this one's got a very odd name. It's the serine hydroxymethyl transferase. Okay, because if it goes backwards, it's transferring the hydroxymethyl group to make serine. Okay, however, we utilize it going the other direction to make glycine. And now that hydroxymethyl group is being transferred to make the methylene tetrahydrofolate and then, of course, water. Oh, I just love it. I don't know why we keep having some issues with the connectivity today.
So would we need to know that like folate goes to methylene folate, or just the enzyme requires the tetrahydrofolate? So would we need to know just the tetrahydrofolate, or what it goes to the end?
You would need to know what it goes to.
Okay.
Oh, come on now. This is really not wanting to work with me here. Let's just go. All right, so now this is really important. I want you just to know the basics from this, because this is how bacteria and plants. But I want you to be able to compare it to the way, since this is a potential spot for an antibiotic, I want you to be able to compare it to the way that mammals make cysteine. Okay, so this is one of the times that I want you to be able to just look at it. First off, let's see. You don't. You just need to be able to talk about like the compare and contrast the way, like where does the sulfur come from? Because sulfur is an element that we don't. So sulfur is one that we don't usually talk about a lot. And so if you notice here, there's several steps for the plants and bacteria to make cysteine. They make cysteine from serine. So that's where the backbone of the amino acid comes from. So I'd want you to know that. So for example, the backbone is serine. But where does the sulfur come from? And on this picture here. It comes from sulfate. Okay, so it uses like the inorganic sulfate salt, okay, as the sulfur donor. Okay, and then like I said, there's several different steps to it. But I do want you to know the way the mammals do. I mean, we only have 2 steps. Okay.
Because we're simple.
We're simpler than that, yes. Okay, so it comes from serine, but with the plants and bacteria, it only came from serine for the backbone. Here, though, ours starts from serine and this player right here, homocysteine, which I've mentioned a time or two before, and this is one you should know the structure for. Okay, because it's homocysteine, and homocysteine has lots of implications, especially in mammals. Does anyone know where do we get homocysteine? We can make it in our body, and it does not come from cysteine. What does it look an awful lot like? Another amino acid. It's methionine that has been demethylated. That terminal methyl group that was before methionine is a sulfide, an RSR. The methyl group gets removed and becomes homocysteine. And then homocysteine, so for us, we make cysteine from methionine and serine. Plants and bacteria only make it from serine, and they get the sulfur from sulfate. We get our sulfur from methionine. All right, so we have homocysteine, we have serine. We use pyridoxal phosphate to remove the water. And so what class or classes of enzymes could this be? Because we're going to be making cysteine. This is and I don't expect you to memorize that name. I would give you the name. And the structure for that matter, because it's quite complicated. But if we're making cysteine using a PLP, what class or classes of enzymes could this be? Is it going to be a synthetase? Synthase. Okay. This one is cysteine synthase. Right, at the beta position. Okay, so now we use PLP to deaminate and whenever that happens, not only do we deaminate it, but we break it apart into two different things: cysteine and alpha ketobutyrate. Okay, so it's a ketone type of body, which can then be utilized to go on for for energy sakes if you need to use it for energy. Okay, so what class or classes of enzymes? This is a class of enzymes that we don't utilize, we haven't talked about a lot. Where it takes a water, it usually breaks things apart. This one, I mean, that's what, usually I would say, oh, this is a deaminase because it's removing the nitrogen, but it's not just removing the nitrogen, it's also breaking things apart. You could try to say hydratase, but that's not quite right either because the hydrogens are not being put directly into it. What? It's a lyase. It's a lyase. Like I said, we don't use lyases very often. This is cysteine, and it's a lyase. So we've only seen that a time or two, in the glyoxylate shunt, whenever plants can break apart using the water. And so this is the part that I wanted you to be able to compare and contrast and talk about the homocysteine. It comes from methionine. S-adenosyl methionine, which is often called SAM. Sometimes they also call it adomet. So let me write that down, because I usually don't call it that, but sometimes you'll see it referred to as adomet.
Sam.
SAM, S-adenosyl methionine.
I don't met something.
I thought so too. I thought it sounds like a King Tut kind of thing. Something from the Mummy. So adomet, but no, it's Sam. So the Mummy's trying to assimilate in our culture and just go by Sam. Okay. All right. Yeah. So homocysteine actually has its own three-letter code. It's HCY. It's prevalent it's important for methyl transfers because methionine a lot of times is used to transfer methyl groups. And so then the product would be homocysteine. It is linked to a whole host of medical disorders. There's homocysteinemia and then homocystinuria. What's the difference what's homocystinuria? It's got so much that's coming out in your urine. And homocysteinemia, pardon? Right. Okay. And so the person who actually ended up going and becoming part of the highest honors for scientists in the United States that you can get is to be on the National Academy. And so it's sort of like a science center, whatever. There's the National Academy and there's the Institute of Medicine, and they usually work hand in hand. They are to advise Congress and the president and other advisory boards, and you have to be nominated. I mean, it's like being knighted into science. The woman who discovered the medical implications of homocysteine collaborated with me. She was actually on not with me, collaborated with my lab. She's actually on my defense. She was on my PhD panel. Her name is Rowena Matthews. I think she's still alive. She's older than dirt, probably. Oh, great! That's gonna go on video too. No, she's great. She's an amazing woman. But yes, come on YouTube. But no, she was named first to the Institute of Medicine, and then also to the Academy of Sciences. And she's an enzymologist that discovered the link between homocysteine and how homocysteine levels rise up and is linked to cardiac stress and cardiac disease. And so they actually they help to come up with ways to test it. And that's one of the things they now test for if you have a history of heart disease in your family, is they look at your homocysteine levels. And so some of these enzymes that are involved in that there are mutations now that are linked to some of the times when you hear about people dying, suddenly from some of the some cardiac issues or things like that. And that all kind of stemmed from the research that she did in her lab. And it was literally like right around the corner from us whenever I was at Michigan. But since then, when they built the new institute, Life Science Institute, her lab moved over into the zoo. That's what we called it. And we stayed back in the lab that they had built for us. But yeah, homocysteine is very, very important. Okay, this is actually from chapter 18. And I want you to know the normal functions. This is just to review a little bit about what SAM does. And so this is just a review from before. We have methionine, and it gets adenylated at the expense of an ATP. Okay? And so it's an adenosyl transferase. That's the first enzyme. And it's one of the only ones that, if you notice, it kicks off a triphosphate in the form of inorganic pyrophosphate and inorganic phosphate. So remember, that was sometimes different than everything else before. Then we have lots and lots of methyltransferases. So those of you who've taken chemistry of cancer, we've talked a lot about methyltransferases in the past. We've talked a little bit about it in neurochemistry too. But there's methyltransferases are used a lot, not only by us, but also by bacteria and other organisms to help dictate which genes get turned on and off, and so on and so forth. And so a lot of them use SAM. The product from that is S-adenosyl homocysteine. Then a hydrolase comes in, kicks it off, and that's where homocysteine comes in. And then we have tetrahydrofolate. And this is where the methyl tetrahydrofolate can then become tetrahydrofolate regenerated coenzyme B12, and you get methionine back. And so one of the ways that you can have heart issues is if one of these enzymes along the way gets out of whack, and then what happens is your homocysteine builds up in your system. So, okay. At this point in time, I'm actually going to end rather than us get started with alanine and aspartate, because we've actually done that in the past.